Hey, what is going on, my peoples? Yeah, I am excited because it's February 22nd, 2022. February, as I've said before, is rolling along and it's steamrolling along. We're getting to the end of it. 6 days left. February is a short month, remember. And we will be reaching. Now we are currently at the cusp in my lecture series, at the cusp of the actual mechanism of cellular growth. Okay, I'm not ready yet to talk about this. In fact, there's quite a lot here, and I will be more ready to start delving into it tomorrow. But today, I'll just give you a little taste. There, we want to kind of understand how cells grow, and that's why I started talking about the different phases of cell division and the cell life cycle, which you start learning about in biology, in intro biology, but then as you start delving into the actual mechanisms that initiate the phase transitions, that's when things start to get very complex very quickly, and they usually cut that part off and don't even save that for college classes. Like, I think a lot of this, you start in cell biology, but then by the time you're in a lot more of it, you go into graduate studies and you start to learn about the details regarding the different chemicals involved in this process. And it's all a very chemically induced process. But we'll go into it. The good thing is we're still in the territory of the known. That means that a lot of this knowledge is exposited somewhere, and it can be researched and compiled and put together. And you notice that I have just to note a search for G protein coupled receptors here. This is what I'm going to talk about, because we want to talk about the growth factors. That's what I'm going to start on tomorrow, probably. But for now, we're just going to go back, and I'll pick up where I left off on the cell cycles, right? The actual cell life cycles and the sub cycle, or the sub phases of mitosis. And where, again, so much of this is interesting to me because it's very complex what's happening here. I can talk about what happens in anaphase, right? Chromosomes break at centromeres and sister chromatids move to opposite ends of the cell. So just to remind you, I left off at metaphase, and metaphase is pretty much at this point when the cell's chromosomes, all the chromosomes are lined up at the center on the metaphase plate, what's called a metaphase plate, and then the microtubules, the polar microtubules, are attached, so spindle fibers as they're called here, to the chromosomes, right, at the metaphase plate. So then anaphase would occur afterwards, right? But not right away. Between anaphase and metaphase, there is a checkpoint which occurs called a metaphase checkpoint. So cellular checkpoints, I haven't even talked about them really, but you can, if you look at this very stereotypical cell cycle diagram here, cellular checkpoints occur within the cell cycles themselves. Okay, so within metaphase, within the G2 to metaphase transition, within the G0 to S transition, there's different checkpoints which occur, which determine if anything went wrong up to this point, right? So that's important because all of this cellular, all these cellular division mechanisms and cell life cycle mechanisms have been going on for eons, right? But they've been perfected over time, and they still, as perfect as they are, as great as they are, they still are prone to mistakes and errors in just manufacturing errors, right? Like just random variations can cause a cell to be set up in a bad way for replication to continue, and we know that's bad, right? So because if cell proliferation happens in the wrong way, bad cells will proliferate, and that's essentially, what happens in cancer, right? With cancer. But that's why these cellular checkpoints are here. So this is basically what these all do. Like I was just talking about the metaphase checkpoint, which I'll get back into. Well, I'll start here. They check for chromosome spindle attachment. So remember, in metaphase, if you can remember, all the cells are lined up all the chromosomes are lined up at the centers of the cells, and anaphase is where the centrioles pull them apart, right? Or via their kinetochores. So what we want to know is that are the cells all lined up at that point, meaning, or the chromosomes all lined up properly? If they're not lined up properly, then what could happen is that some of the chromosomes will get incorrectly pulled or not even pulled at all, and then you could have a cell with an incorrect number of chromosomes at the point where its telophase is about to begin and the cells pinch off into separate daughter cells. So we don't want cells with deformed cells, essentially cells with bad DNA or incomplete chromosomes within the cell. So at that point, I can imagine, I don't know for sure, but some sort of apoptotic process could occur, which would cause the cell to die, or would cause a T cell of some kind or natural killer cell to come by and eliminate that bad cell. I don't know if that occurs, or maybe the entire cell division stops in both cells. The entire potential cell dies at that point. I'm not exactly sure how the body decides yet. I'm sure there is a process. But anyway, so that's what happens at the cellular checkpoint, metaphase cellular checkpoint. At the G2 checkpoint, we have a check for cell size and DNA replication to make sure the DNA replicated right before going into metaphase. I'm just mentioning this because I'm going to go into telophase next, obviously, but then at the G2, there is the G2 checkpoint before metaphase, or before mitosis, to make sure that mitosis can occur. So we have to make sure the cell after G2 has the right amount of DNA, and we have to make sure it's the right size, because otherwise there won't be enough room. The new cell won't have enough room for all the organelles. The cells, when they divide, they're supposed to have exact copies of everything in there. Not exact copy, but they have to have the same number of organelles, same number of ribosomes, same amount of DNA. Certain items have to be accounted for to a particular number between both daughter cells. And if that's not the case, then again, these cells probably eliminate themselves. The G1 checkpoint, that's after the cell goes into, that's the point at whether the cell can go into the growth resting state, or it can go into DNA synthesis, which means it will go through another cell cycle. So we have to determine whether the cell is ready. And I believe at this checkpoint, the G1 checkpoint, the cell, it's less all or nothing, in the sense that the cells probably just check whether they're ready. They don't necessarily kill themselves at that point, right? So this is the least deadly, I guess, checkpoint for cells. But I'm not exactly sure again, but it seems like that's the case. So let's just jump back. Those are the checkpoints, and at those checkpoints, there's so much going on, it's unbelievable, but we will talk about that later. That's all part of cell cycle regulation, and that's, I was delving into the stuff about the G proteins at that point. So let's talk about telophase. So remember, after anaphase occurs, right? Anaphase occurs. Anaphase is when the chromosomes get pulled over to the microtubule organizational centers, right? And then in telophase is when the nucleus starts to reform around the cells, DNA, right? The individual cells. So you start to see two nuclei, right? You have all the duplicate endoplasmic reticulum, duplicate ribosomes, duplicate ribosomes, duplicate mitochondria, and you have the cell looks like it's starting to become two cells, right? So what happens, there's this pinching off that occurs, this constriction ring that occurs between the cells, and then at that point, cytokinesis, which is a part of telophase, occurs, right? So this contractile ring appears, or cleavage furrow, as it's biologically referred to by many scientists. The nuclear envelope, again, is here more distinctly prominent, and then the pinching occurs. Many proteins and enzymes are involved in this entire contractile process, the ancient contractile process, and then the cell becomes two different cells. And at that point, the cells can, depending on what the need is for their proliferation, they can either go into the G1 phase again right away, right? And that means start growing again to divide. Epithelial cells, labile cells, hematopoietic stem cells, all sorts of cells that need to proliferate frequently go back into the G1 phase and start to grow again in the prospect, or for the prospect of another cell division. Otherwise, they go into the G0 phase, and the G0 phase is the phase at which the cell is called a resting phase. The cell cycle stops. Now, the cell cycle in the when the cell is in the G0 phase, it can be termed as most commonly quiescent, meaning it's not doing anything except its normal cellular operation. The whole point of the cell, it's not to divide necessarily. The cell needs to divide, that's a fundamental function, but it needs to do other things, like a blood cell needs to carry blood, right? Other cells need to do other things, produce the right chemicals, so they'll be doing their work in the G0 phase, right? But in the G0 phase, there's the quiescent version of that, and then there's the senescent version of that, where the cell is stuck at the G0 phase for all of eternity until it dies for whatever reason. And that's where we want to get to, because cellular senescence in life extension is and you've probably heard this if you're familiar with longevity studies, is one of the most important aspects controlling cellular senescence, right? Because senescent cells are senescent for a reason. They've accumulated so much DNA damage that they can't they can't function, and therefore they have a chance of becoming cancerous if they divide again. So cellular senescence is a mechanism that's there to stop those cells from dividing again. So yeah, and what we'll do next is we're going to talk about everything that's involved with controlling the cell cycle and the regulation of the cell cycle. Not everything. There's so much involved. We'll touch on the surface, but we'll talk about proto-oncogenes, tumor suppressors. We'll go back to the G-coupled proteins and growth factors, because G-coupled proteins are integral to the cell cycle regulation that indicates a cell should start growing. In cancer research, oncogeneticists, they know very well about the details involved in many of the G proteins, and many of the G proteins are not known. So I kind of jumped over the specifics of telophase because cytokinesis, all that stuff, that's, again, the level is high school biology, and of course we're going to go back into each of those phases more specifically. This is where we're going to, the brain space, this and cognition are where the brain space of these videos is going to primarily reside in the near future, at least, and the immune system. So those three areas, because they're all what I believe to be, at this point, the most important pieces in human cellular biology to understand if we're going to make any progress towards the progression of life, eternal life, etc. Okay, so that's it for this video. I hope you had a informative time, and I will see you in the next one. Thanks.